So hello guys and welcome back to Excel Academy. So today we'll be doing the last topic, that is transcription in eukaryotes. Okay, so I have mentioned before itself like what are the transcription factors, like as in the case of the prokaryotes, those many proteins were not required, correct? If you remember, we have discussed about the sigma factor which is coming and recognizing the -35 and -10 promoter sequences, correct? Now in the case of the eukaryotic transcription, again the promoter sequences are known as data box, and in this case there are many proteins or many transcription factors which is involved in order to form a initiation complex. So slowly we'll go to that, and there are many interesting concepts about like how the transcription is happening. Like the first question should come like is that whenever we talk about any transcription, we always say that the DNA is there, it gets opened up and and the mRNA is formed, right? But we need to always remember that your DNA is always not free. What does it mean? Because it is always bound with the nucleosomes. Okay, now these nucleosomes are making sure that your DNA is not free. तो हमें जब भी transcription कर रहा है, we need to remove these nucleosomes and then only we can do the transcription. Okay, so this is also one important topic in eukaryotic transcription which we'll be covering it today. Okay, so as we said, there are 3 RNA polymerases. So we have got RNA polymerase 1, 2 and 3. जिसमें से 2 है जो आपका mainly mRNA के लिए synthesize करता है, and then we also have got the other types of RNAs like small interfering and the microRNAs. Okay, so these are also playing an important role in the defense mechanism in the many cells. Now coming to the RNA polymerase 1 and 3 nucleolus, can you see the difference? ये nucleus में होता है, ये nucleolus में होता है, and then it is coding for your rRNA. Now last one is for your tRNA and other small RNA particles. For example, the signal recognition particle, ये हम कहाँ use करते हैं? Any idea? Have you heard about this signal recognition particle? All these SRP RNA, they are used in the spliceosome. अब spliceosomes का क्या काम है? It helps in the intron splicing. Okay, so after transcription, the another topic is about the RNA splicing. So we know that in the case of DNA, the introns are also present, correct? And then we have got the exons. So when mRNA is formed, everything will be present in this. Okay, mRNA, everything will be there. Later on, with the help of these spliceosomes and the ribozymes, these introns will be removed, and finally a matured mRNA having only the exons or the coding sequences will be present. Okay, so far so good, guys. Able to follow?
Yes.
Okay. The thing is, the complexity is going to increase. Okay, so just let me know if I'm going fast or not able to follow. From here onwards, you need to remember so many proteins name and that way in order. Okay, so this is how the promoter of RNA polymerase 2 looks like. Now this, if you can remember, then it is easy to remember which all transcription factor is coming and binding. Okay, so let's see the promoter over here. You don't have to remember the sequences. This is not required. Okay, so these sequences you don't have to remember. Okay, so you can see that here also you have got the data box. Okay, so the data box over here is your TF2B, the BRE. So BRE is your TF2B recognition element. B stands for TF2B, RE recognition element, which is just next to your data box. Now what is TBP? This is data box binding protein. Okay, binding elements or binding proteins, they say. Clear? Now this is initiator. I and R is your initiator. Now apart from in the prokaryotes, in case of eukaryotes, we have got the other mediator proteins also, which is helping in the formation of the PIC. What is PIC? Pre-initiation complex. Okay, so इसमें हमारे पास है initiator also, we have got enhancers also, and the last but not the least, the activators. Okay, now please do remember, in the case of eukaryotes, we do need, along with the transcription factors, initiator, enhancers and the activators. अब नाम से ही पता चल रहा है कि इनका क्या function है. Initiator to help the initiation, in the formation of the initiation complex. Enhancer क्या करेगा? It gives a push to the RNA polymerase, and the activators are helping in removing the nucleosomes, because it is a chromatin model, right? Your DNA, it is a chromatin, it is not a free DNA. So over there you have got the nucleosomes present. So these activators are going to help in removing the removing the nucleosomes, carrying out the transcription and bringing back the nucleosomes back. Okay, now, so these are the initiators to which your transcription factor 2D is going to bind and will start in the helping in the formation of the initiation complex. Okay, now you can see that we have got the DCE and DPE elements. So what are DCE? They are the downstream promoting elements, and then downstream core elements are there. Okay, nothing but the sequences which will be recognized by the other transcription factors. Okay, so far so good. So you don't have to remember the entire promoter of the RNA pol 2. You just need to remember that it has got the BRE sequences, that is your TF2B recognition elements. It has got the Tata box, that is your promoter. Then the initiator, DCE, that is your downstream core elements, and downstream promoter elements, DPE. Okay. So if you could remember this, more than enough about the core promoter of the pol 2, RNA pol 2. So far so good, guys. Can I proceed? Yes. Huh? आगे बढ़े? Okay.
Yes, ma'am.
Now, this is the same figure I have given in the last lecture, so that you get an idea. ठीक है? अभी धीरे धीरे हमें समझ में आ रहा है. So you have got the promoter. So TBP comes over here and TF2D is there. So your first transcription factor which comes and binds the promoter is your TF2D. Okay, along with the TPP. Then comes your A and B. So I hope you remember the sequence. बहुत जरूरी है sequence याद रखना कि like in which order the transcription factors are coming. Okay, so remember it is TF2D, and don't write 2 like this. It is always like this. Okay, so TF2D. Okay, then plus TBP. You can make a short notes like this. You don't have to draw the entire thing. Then comes your A, then comes your A, then comes your B, then F. Now here also important thing is that A and B binds to the promoter sequences, whereas F. So this F is actually recruiting the RNA polymerase 2. So you can see over here that it is bound to the RNA polymerase 2, and then it brings to the promoter region. And then comes your E and H. Okay, so this is now completely ready. So this is your pre-initiation complex. Now later on, the tail of the RNA polymerase, okay, the tail of the RNA polymerase is getting phosphorylation. Now here also very important to remember the number of the phosphorylation. Okay, so at which amino acid it is getting phosphorylated, the RNA polymerase is going to have difference in the function. Okay, it will tell you that whether this RNA polymerase is at initiation stage or elongation stage or termination stage. So this phosphorylation decides the stage of your transcription. It will also tell what next step is going to happen. Okay. Okay. Now, before I proceed, I want to ask one small thing. So you can see over here that the mRNA has start coming out. Okay. I mean, I will be saying mRNA only, aadat hai, but since it is RNA polymerase 2, so we'll be talking about mRNA. Okay. Otherwise, we should go in a generalized like RNA. Okay. So you can see that here mRNA is there. Now, tell me how the system, okay, the cell transcription system is going to make sure that this mRNA is not going to get chop off. I'll tell you one thing, in the nucleoplasm, you have got the nucleases present, okay? You have got the RNases present. So the moment they see that there is a free mRNA, what they are gonna do? They will start chopping it. Because क्यों ऐसे सिखाया है? They are programmed to chop off the mRNA because it can be from virus also. So if they see that there is a free mRNA floating in the system, they are going to chop off. Okay, but this is the mRNA of the cell itself. वो हमारा खुद का है. So how I'm going to prevent this attack? Yes. Any thoughts on this?
While producing the RNA itself, it is tagged by self proteins or something, so that the nucleosomes or whatever know that this is not supposed to be eaten. There must be some kind of a tag that they use.
So what is that tag? We all know that tag. See, it's okay. गलत हुआ तो क्या हुआ? So it's true that every time the cell has to protect itself. Okay, and for that there is a tag. The another name of the tag is cap. So now what is the answer? So you have heard about the 5' cap, the capping system, correct? So so when we talked about, see हम लोग स्कूल में कैसे पढ़ते हैं, sorry not school, colleges में कैसे पढ़ते हैं that पहले transcription हो जाएगा, whatever mRNA comes, and there is the modifications which is happening in the mRNA, that is your 5' cap होगा, and towards the end there will be addition of a poly A tail होगा, correct? But this is not in the system. It is happening कि पहले आपका transcription complete होगा, उसके बाद capping होगा, उसके बाद poly A tail होगा. No, it has to protect itself from the nucleases and the RNases attack. Okay, so simultaneously this is happening, and this signal is given by the type of phosphorylation happening on the RNA polymerase 2 का tail. Okay, able to hold on to the information? समझ में आ रहा है? तो जैसे जैसे आपका RNA polymerase is elongating, okay, the phosphorylation over here is changing. Now that will decide कि now I should recruit the enzymes which is required for the capping of the mRNA. Okay, since it is exposed now in the nucleoplasm, right? So in the nucleoplasm we have got these enzymes which will chop off if it is kept unprotected. Abhi clear hai? Okay, shall we proceed?
Yes, ma'am.
Okay, so we'll summarize it also. I'll give you the brief picture. I didn't get the proper video of this. I really tried, which will give you the entire, the brief video or a brief summary of the transcription. What I got is like everything which has they made using the PowerPoint only. I don't want that. Okay, so this is the example of the activator. So you can see how these activators are bringing they're just making the loop over here and bringing the chromatin remodeler, mediator complex, and the hat. Okay, so all these proteins the activator is bringing so that it can now open or remove the nucleosomes. Okay, now before I proceed, so what is hat? Any idea what is hat? Hat and what to say HDAC, you must have heard about it. Yes? Nahi? Okay, no problem. It's okay. There's so much to grasp over here. So hat is your, see from the word itself, you can see that we have got the chromatin remodeler. Okay, chromatin remodeler means they are helping in remodeling the chromatin structure. But when you talk about remodeling of chromatin structure, you can see that the nucleosomes, okay, those what are those nucleosomes? They are nothing but your histone proteins, correct? Okay, so we also know that these histone proteins are octamers, so they need to be removed and realigned. And to remove, okay, hat, what does hat is doing? It is nothing but your histone acetyl transferase. So from the word itself, you can understand that it is going to add the acetyl group to the histones. Okay, And please remember, in the histones, so histones are also your positively charged proteins or basic proteins, correct? So they have got increased number of lysine and arginine. So these histone, these hat proteins, enzymes, they are going to add this acetyl group to the lysine of the histone. Now, since it is positive charge, your DNA is negative charge. So there is a interaction happening between the nucleosomes and the DNA, and it is beautifully wrapped around. Okay. Now, to this positively histone charge, when I add acetyl group, what is acetyl group? CH3COO minus. So when I add acetyl group to the histone, what is happening to the histones and the interactions with the DNA? क्या होगा तब? Simple chemistry.
Ma'am, it will lose affinity with the DNA because it will become neutral.
Yes, absolutely correct. So when you add acetyl group, so histone का जो positive charge है, that will be neutralized by the negative charge of the acetyl group. Now, because of this, it is going to lose the interactions with the DNA. So now what is happening? The histones are leaving and the DNA is free of any other proteins. So अभी DNA आपका free है, where the RNA polymerase can start its transcription. ठीक है? अभी clear है? So this is how it does. Now what you need to remember over here is that एक question ऐसी पूछा था, I just remember. So when acetyl hat is adding acetyl group to the lysine, what happens to the lysine? Okay, so it's just nothing that if you remember the structure, it goes to the last group of the lysine to form a epsilon N acetyl lysine. ठीक है, इसको हम ऐसे बोलेंगे. So if you know the structure of the lysine, then it becomes easy to understand. Okay, so how is the structure of the lysine? Anyone knows it? कोई बोल सकता है? So here you have got the, let us say that this is NH2CHCOOH. ठीक है, तो ये हो गया आपके नॉर्मल जो आपका अमीनो एसिड का रहता है. Then the side chain. So CH2 simple है, 4 बार CH2 है, and then last you have got the NH2 lysine. So it has got the amino group. So this is known as your epsilon. तो यहाँ पे H हटा के you have got the acetyl group. ठीक है, so that becomes your epsilon N acetyl lysine. Simple, clear, याद रहेगा. Now why it is epsilon? If you know the nomenclature in the amino acids, from here, okay, the chiral carbon will start from here: alpha, beta, gamma, delta, and epsilon. Okay, clear? So epsilon ये carbon हो गया आपका, then to this epsilon carbon this is attached. Okay, clear? Any doubts? Is this concept is clear about the activators, how they are helping?
Yes, ma'am.
Okay, सब कुछ याद रखना है, ठीक है? Don't worry. I mean, we'll be doing the revision also. Now, so that was how your pre-initiation complex started, and now एक और चीज याद रखना है, as I mentioned before. So there will be a, coming as we discussed in the case of the prokaryotes, that RNA polymerase जाता है, it makes few nucleotides, then again comes back and then again starts. ऐसा यहाँ पे नहीं होता है in the case of the eukaryotic transcription. Okay, for that over there sigma is there. If you remember, we have discussed about it, that it makes a few nucleotides and comes back and then again starts. So once it reaches around 20 nucleotides, then the sigma lays and the elongation happens. So such things are not required in the case of the eukaryotes, because already those enhancers and the other transcription factors are making sure that the RNA polymerase continues the transcription once it is initiated. Okay. Now let's talk about the elongation and the RNA proofreading. So once the polymerase has escaped the promoter and initiation shuru ho gaya, it is going into the elongation phase. Okay, obviously. Now we have got these transcription factors and the mediators, the enhancers, activators helping in order to carry out this process. Now there's another set of factors which is recruited. So these are known as the elongation factors. Those are transcription factor 2S and SPT5. Okay, abhi confusion nahi aana chahiye. So already we have discussed about the initiation factors, correct? Now these are your elongation factor. Okay, SPT5 and TF2S. Now, what is the activity of this TF2S? It is to stimulate the transcript cleavage, okay, which in the case of the, if there is a arresting of the RNA polymerase 2. समझ में आया? अगर आपका RNA polymerase कहीं अटक गया है, तो it needs to free the transcription, okay? So your TF2S is present. Clear? Yes, guys, able to follow? See, there might be some problem in the DNA because of which your transcription gets stalled, right? So in order to make it clear, आपको आगे बढ़ने के लिए, so this is going to help in the freeing the transcription, freeing the transcript, okay? Now, the next one, SPT5. So what are these? They're also, they act both as positive and the negative regulators of the transcription elongation. Okay, so they help in the chromatin remodeling. अभी आपको ये याद रखना है because we are going to talk about the chromatin remodeling also, where this protein is going to be used. Okay, so can you just remember this much? Like in the case of the elongation process, in the transcription of the elongation, there are two factors are there. Those are your TF2S and SPT5. Okay, clear? Can you remember this much? So what is the function of TF2S, Tanvi?
Ma'am, when RNA polymerase gets stuck somewhere, so TF2 will come.
Yes, to free
elongation.
Yes, very good. So that the RNA polymerase gets free and it can start the transcription again. Okay. Now, what is the function of SPT5? Okay, I haven't mentioned. I just said that the chromatin, it is helping in the chromatin remodeling. So इसके पास दोनों function होंगे of being a negative and a positive. क्यों? First, you need to what to say, chromatin remodel करना है, and then again you need to bring the chromatin in the same position, right? Same वापस उसी position में लाना है. Otherwise, what will happen if you have got open DNA? Now, those open DNA is like a targeting site by the other nucleases. Correct. So that will be wrapped around by the nucleosomes and help with the SPT5. Okay. So far so good, everything. And recruited to the CTD tail of the last subunit of Pol 2. Okay. So we know that the tail of the RNA polymerase is bringing these elongation factors. Okay. So here done with your elongation. Now look at over here. Very, very important. Okay. So you can see, if you have got no phosphorylation, okay, no phosphorylation in the tail of your RNA polymerase, it is your pre-initiation step. Okay. Now, if at the fifth position, the serine is getting phosphorylated, and always remember it is the serine which is getting phosphorylated. Okay, no other tyrosine or any other protein. So we know that the tyrosine is also having a OH at the end, and serine is also having the OH. But it is the only serine which decides the fate, okay, or the stage of the transcription. Now, if you see in the figure, so if you see it over here, you can see that if there is no phosphorylation, it is in the pre-initiation, okay. Now, if it is at the fifth position, serine, then the promoter escape is there, and now it is going to recruit your capping enzymes. Clear? Okay. Next, if it is at the second position, serine is getting phosphorylated, it is in the elongation step, and the splicing will start. And for the splicing, your cleavage factors, those are your signal recognition particles and other SNRPs, we say SNRPs. Okay, we'll talk about this later. So all these proteins will be recruited so that जैसे जैसे आपका mRNA बनता जा रहा है वैसे वैसे introns will be removed and only exons will be present over there. ठीक है? Clear? And of course the poly A tail adding proteins will also be recruited. So guys, clear? This figure is important. Okay, you need to remember this figure. and the positions of the serine getting phosphorylated. So let us just revise once more. So if you don't have any phosphorylation, it is in the initiation. Fifth position, serine is getting phosphorylated. It has left the pre-initiation and now it is present in the elongation, where the first enzyme which it has recruited is a capping enzyme. Next, if it is in the second position, it is in the stage of the elongation and later it is going to recruit the splicing proteins. Okay, clear? Shall we move ahead? एक बार देख लो. If any other doubts are there, then we'll proceed. Okay. Shall we proceed?
Yes, ma'am.
Okay. So now the another protein. Okay. So these are the different proteins again which is helping in the elongation. Okay. So one of those is the kinase TEFB, which is recruited to the polymerase by the activators. Now, once this protein is bound to Pol2, the protein phosphorizes serine residues at the position 2 of the CTD. So now we know which is that protein which is doing the phosphorylation at the serine. So that is your kinase P TEFB. Okay, please remember ये एक बार आ चुका है एग्जाम में. Kinase P TEFB. Okay. Now, in addition, PTEFB phosphorylates and thereby activates another protein called SPT5 and TAT-SF1. Okay, so we know about this now. Now, SPT5 का काम है 5' cap के लिए and TAT-SF1 का काम है for your poly A tail. Okay, so what is the takeaway message from this slide? So the Pol2 का tail जो है, that is your carboxy terminal domain, it is getting phosphorylated at the serine residues by the protein called kinase P-TEFB. It also recruits your SPT5 and TAT-SF1, where SPT5 is responsible for your 5' capping and TAT-SF1 is responsible for your poly A tail. Clear? Simple, right? Okay. If you want, you can go through with the like additional topics about like additional you can read from the Google, because in the text it is not given much. In the text only this much is given, that it is a kinase PTFB which is helping in the phosphorylation at the RNA Pol2 tail. Okay. Now, so what is FACT? This is what we were talking about when we were saying about the chromatin remodeling. Okay, so now let's talk about the chromatin remodeling. So what is your okay, so here about the FACT. So FACT stands for facilitates chromatin transcription. Okay, so as the name is suggesting, it's a factor which helps the transcription on chromatin template much more efficiently. So it is a heterodimer of a protein formed of SPT16 and SSRP1. अब वो आपके ऊपर है कैसे आपको याद रखना है, and I'm very much sure that these are confusing. Okay, just now we talked about SPT5 and SPT16, ये सब हमें confuse करते हैं. I do agree, but I don't know how to tell, but this is how, like what I used to do is I used to make as a column wise, like FACT means these proteins. Like you can have your own acronyms also if you want. Okay, so nucleosomes are optimus. We know it is made up of H2A, B2, B3 and 4. So these are 4 into twice. Okay, and we know that 2A forms a dimer with the B and 3 forms a dimer with the 4. So SPT16 it binds to H2A and H2B. So SPT16 आपका यहाँ bind करेगा, and to the H3H4 your SSRP1 binds. Okay, so what they are going to do is they dismantle the histones by removing the dimer, one of the dimer, and then again reassembling. So similarly for your H3H4 also. Okay. Excuse me. So this removing of the histones will allow the polymerase to pass the nucleosome. And once it passes, FACT is also having a chaperone activity. Now, what is chaperone activity? That is bringing the protein together and helping in formation of the structural complex so that the nucleosomes are formed again and back to the normal. Okay, so FACT allows polymerase to elongate. At the same time, it also maintains the integrity of the chromatin through which the enzyme is transcribing. Okay, so you have got the chromatin like this. The moment yahan ka nucleosome is open, RNA polymerase will pass and your nucleosomes will be formed again. Okay, so it makes sure that this is happening. So of course, so many FACT molecules are involved in this. It's not like only 1 or 2 nucleosomes will be open, right? Many nucleosomes will be open so that the entire gene gets transcribed. Is this much clear? Yeah, I know so many protein names are there, it's a bit confusing, but over a period of time, if you listen 2-3 times to the video or make a short note of it, will be easy. Okay, so till now we were talking about the chromatin rebonding. I'm just summarizing the slide. So there are certain proteins, certain factors during the elongation, which is helping in opening of the chromatin nucleosomes. Okay, so we know that the nucleosomes are made up of octamer of H2A and H2B, iska tetramer, and H3 and H4 ka tetramer. Okay, so to this your spt16 opens, and to this your ssrp1 opens. So they help in the opening of these histones, and then later once the RNA polymerase is passing, it will form back to the normal nucleosomes or the chromatin structure. Guys, easy to understand. It's clear, right? Yes. I mean, I do know that you might be not able to remember all, but I hope the concepts are clear. Like, kaun kya karta hai? That is clear, right, Tanvi? If any doubt is there, I can repeat. Yes, anyone? Shruti, Akshaya, is it clear? Okay, हाँ, that's what is required. क्योंकि एक ही बार में सब कुछ याद नहीं होगा. You need practice. Okay, you need to remember. It will take time, so it's okay. You take your time to remember all these concepts, but of course before the exam. Okay. Now, this is the same figure जो भी हमने बात किया. So you can see it over here. See, SPT16 and SSRP1 is there. Oh, sorry. So this is, they are helping in removing these octamers and then again bringing back, correct? And then RNA-P polymerase is there, it is moving ahead. जैसे पीछे का हो जाएगा, they start forming back the histones. And then again you can have the SPT16 and SSRP1 over there. तो जैसे ये open होगा, RNA polymerase will cross this thing and they will come back again forming the octamer. So slowly this will go on till the entire the termination sequences come. Okay, the genes are transcribed. Now 5' capping and about the poly A tail. So as we have already said that this is done by the SPT5. Okay, so this is recruiting your 5' capping enzyme. Okay, now you can see it over here. ये आपका top of RNA है, mRNA है. So you have got the triphosphate over here. So your first, sorry. So your first one is going to be the triphosphate. क्योंकि उसके बाद ही you start having the other nucleotides, which is there in which you can have only the one single phosphate forming the phosphodiester bond, right? Now, so this is having the serine 5 position phosphorylation, right? Remember, so SPT5. Now you can see that the 5' capping enzyme, how it is doing. So it is going to bring, which is your 5' capping enzyme? Those are your guanyl transferase and methyl transferase. So what is happening? Guanyl transferase kya karega? It is bringing your GTP. Okay, it is bringing a GTP. Now GTP is coming and you can see that the beta phosphate, okay, the beta phosphate is attacking over here. Okay, and then what will happen? A triphosphate bond is formed. Can you see it over here? 5' bond is formed when this is attacking over here. Okay, so 1, 2 or 3, this phosphate and this phosphate and this phosphate will remove, and ये pyrophosphate जो है वो remove हो जाएगा. So you are going to have a phosphate, triphosphate, where in which you have got the 5' and 5' of this, 5' of this. Okay. Then methyl transferase will come and add methyl group over here at the 7th position. So that's how it becomes 7-methyl guanine, 5' guanine cap. Clear? Simple. There are only two enzymes required: guanyl transferase and methyl transferase. Guanyl transferase क्या करेगा? It is going to add a G at the 5' end of your mRNA. Correct? So this is important, that it is forming a 5' phosphate bond. Then methyl transferase at the 7th position, it is going to add to the guanine. So it is also known as 7-methyl guanine. 7-methyl 5' guanine cap. 7-mg also they say sometimes. Okay, is this much clear? There are only two enzymes. आपको सिर्फ दो ही का नाम याद रखना है. That is guanyl transferase and methyl transferase. Simple. Okay. No one is asking to draw the structure, so you can just remember the names of the enzymes. Now let's talk about the poly A chain. So for this also, you need two proteins again. So the two protein complexes which is carried out by the CTD polymerase. If you remember, it is the second serine which is getting phosphorylated. Okay, so that is recruiting your two proteins which are CPSF and CSTF. So CPSF stands for cleavage and polyadenylation specificity factor and cleavage stimulation factor. Okay. So once the sequences are transcribed, okay, जहाँ पे आपका termination sequences आ रहा है, it is approaching those, okay. So these proteins, okay, that triggers the transfer of these factors to the RNA, and those factors, those signal is known as the poly A signal. So you know that at the end of the mRNA, so those sequences will be present. So once the RNA polymerase are transcribing those sequences, the poly A signals gets triggered. And now this trigger will bring your CPSF and CSTF to the RNA. Okay. Now, so this polyadenylation, it is mediated by the enzyme called poly A polymerase. It is your poly A polymerase. Now, this is going to add around 200 adenines to the RNA at the 3' end. अब वो 200 ही क्यों add करता है? We don't know about that. Still the research is going on. Okay, and of course it uses ATP as precursor in order to add, and the same mechanism how it is doing. So it will keep on adding the nucleotide how RNA polymerase adds without, but the only difference is there is no template because it just have to add A only. So it does not need any template, but the insertion of these nucleotides, the activity is similar to that of the RNA polymerase. Okay. I hope you guys are not getting confused. Should I summarize the slide? Do you want me to summarize?
Yes, ma'am.
Okay. So see, simple. So we remember earlier that when there is a second position serine getting phosphorylated, it is going to recruit your poly A tail forming enzymes. Okay, so those are the sequences which when gets transcribed by the RNA, the poly A signal gets activated. Now these signals are going to recruit your CPSF and CSTF. What is CPSF? It is cleavage and polyadenylation specificity factor, and this is your cleavage stimulation factor. Okay, CSTF. Okay, now once these are recruited, they are going to recruit your poly A polymerase. Okay, this has got the activity similar to your RNA polymerase. Only difference is that RNA polymerase needs DNA template in order to add complementary nucleotides to synthesize mRNA, correct? But in the case of poly A polymerase does not need template. Why? Because it is going to add only A adenines. How much? Around 200 we say. So 200 adenine it is going to add. That's all. Simple. 3 sentences में आपका poly A tail formation हो गया है. So which is the enzyme? It is your poly A polymerase. But you need the help of the CPSF and CSTF. Clear? Okay. Now this is a figure. Once again. Okay. Now look at the figure. So once it is getting phosphorylated, all your signals, see CSTF, CPSF, they go and bind to this poly A signal, which was transcribed by the RNA. So we don't want this additional RNA जो transcribe हो गया, ठीक है? So once this is being done, the RNA is free. Okay. So once this RNA is free from this complex, okay, you have got the extra synthesized nucleotides. Can you see it? हमें सिर्फ यहाँ तक चाहिए था, okay, but then we have got this much extra also, so that will be chop off. Okay, now that will be chop off, it will be removed. Then you have got the PAP. What is PAP? Poly A polymerase, and it will keep on adding your nucleotides A. Okay, clear guys?
Yes, sir.
है ना? I know थोड़ा सा है, like we need to remember a lot over here. Yeah. Now coming to the termination. So this is your termination ka slide. Very simple. So they have the researchers have proposed two models for the termination. Okay, so those are known as torpedo. So this is your torpedo, and this is your allosteric model. Now, if you compare with the prokaryotic termination, what was happening in the prokaryotic termination? In the case of prokaryotes, it was all about row dependent and row independent termination, right? If you remember, correct? So what was happening in the row independent? Since there was a complementary base pairing, a hairpin loop structure was formed, which resulted in the skipping of the RNA from the RNA polymerase, right? In the case of row dependent, there was a hexameric protein which was helping in the termination of the RNA, termination of the transcription, correct? If you remember, in the case of the prokaryotes. Now, in the case of the eukaryotes, we have got the two models, which is proposed as torpedo model and the allosteric model. Now, in the case of the torpedo, what you can see over here is that, see, the moment this portion has been removed, okay, can you see the 5' cap, mRNA, poly A signal, हमने ये remove कर दिया है. The remaining whatever is present, the torpedo is going to come and chop off. Okay, so this is how it is going to make sure that the transcription is stopped. Clear? Clear? Any doubt is there? अभी हमने क्या देखा था, that poly A sequences comes and the poly A polymerase will come and it will take all those CPSF and CSTF के बारे में हमने पढ़ा था, right? So they will be helping in removing of the और RNA. RNA जैसे ही हटा, poly A polymerase is coming and starts adding the A nucleotide. So जो whatever was left, okay, whatever was left, it will be chopped by the torpedo. Torpedo is the name of the exonuclease, RNA exonuclease. Okay, for example, here it is present. In the case of rat, it is known as the rat1. One second. In the case of rat, it is known as rat1, and in the case of humans, it is known as HXRN2. It is H human XRN2. In the case of humans, it is known as this. Or in simple word, it is RNA exonuclease. Or other words is torpedo. Clear? So this is finally going to trigger the dissociation of the DNA template and the transcription of the termination of the transcription. Hence, your RNA polymerase is now free to go. Okay, this is one model. What is the second model? So they say that in this case, once the poly signal is passed, the RNA polymerase becomes less possessive, okay, less possessive for its activity. So because of this, there is a conformational change. जैसे आपने RNA polymerase नीचे से निकाल दिया, okay, once after the poly A sequence has been passed by the RNA polymerase, it becomes less possessive. Now, because of this, there is a conformational change, and hence it will be leaving the DNA template strand and leaving. Okay, so जो भी बचा कुचा RNA था, now this will be degraded by the RNases. As I said, RNAs का काम है कोई भी free RNA को chop off करने का. Okay? Any doubts? या नहीं clear है? Should I repeat? Should I repeat, guys? Please let me know. तन्वी clear है क्या?
Yes, ma'am.
Okay. What about others? Shruti and Akshay?
Yes, ma'am.
Huh? So I hope it's clear. The only thing is we have talked about so many proteins today, like starting from the initiation, right? So we talked from the initiation, we have started talking about the so many transcription factors are there. Then in the elongation also there are two more elongation factors. Then in the case of termination, you have got the two models. Okay. Now two models means people have got proof of both. So either it can be with the help of the HXRN2, that is your RNA exonuclease, or it can be because of the allosteric model. Allosteric model क्या बोलता है? That once the poly A signals are being passed by the RNA polymerase, the RNA polymerase becomes less processive. Okay, this brings about the conformational change in the RNA polymerase, due to which it is losing the DNA template strand. और जैसे वो छोड़ देता है, whatever the left RNA was present, this will be degraded by RNases. ठीक है? So this is about the termination. And in the elongation, we have also discussed about like how the FACT is helping in the chromatin transcription. Okay, and we have also talked about the 5' capping enzymes and the poly A tail. So whenever anyone is asking in the exam also, like where does this 5' capping and poly A tail comes, it is not after the transcription, it is during the transcription, that is in the elongation step. Okay, so 5' cap is because of the 5th serine residue is getting phosphorylated at the tail of the RNA polymerase. Poly A tail is because of the 2nd serine residue getting phosphorylated. And now who is phosphorylating all these who is phosphorylating the tail of the RNA polymerase? It is your kinase, Peter B. Okay, so this is the summary. Okay, now I guess we have got few questions. So let's try to get the answer. Kuch hard and fast rule nahi hai. We'll just try to get the answer. Okay, so this is based on what the topics have already been covered. Okay, so your first question is, I guess hardly 2-3 questions are there, not more. So let's do this question. Okay, so your first question is, during development and differentiation, there is a dynamic program of differential expression of sets of genes. Okay, in bacteria, phage infections are among the simplest examples of developmental process. Typically, only a subset of the phage genome, referred to as immediate early genes, are expressed in the host immediately after phage infection. क्या बोल रहे हैं? That if there is a phage infection, उसके जो early genes होते हैं, वो ही आपके host में express होते हैं, ठीक है? So as the time passes, early genes start to be expressed and the immediate early genes and the bacterial genes are turned off. In the final stage of infection, the early genes give way to the late genes. One of the simplest way it is achieved is through dash. Expression of cascade of sigma factors, expression of new RNA polymerase, expression of different holoenzymes, or the expressions of the different transcription factors. So what do you think, what will be the answer? Yes. Question clear hai? So the question says that RNA polymerase, sorry, not RNA polymerase. The question says that after the bacteriophage infection, जो उसके immediate early genes हैं, ठीक है? So once it goes into the host, those are expressed later on. So यहाँ पे host के जो proteins है, जो host के जो genes हैं, those are turned off. But for the phage infection, those genes are expressed. तो ये किस वजह से होगा? कैसे अचीव किया होगा वायरस ने? Yes, Tanvi. Any idea what will be the answer? Okay, let's see what all options can be correct. Expression of cascade of sigma factor. See, I told you this sigma, okay, this always decides what kind of RNA polymerase needs to be active and recruited. Okay, so अगर यहाँ पे आपका हो रहा है the early genes give way to a late gene expression of the bacteriophage, so it means that the different type of sigma factors are being recruited. Okay, so by this sentence, like the explanation I have given, what do you think that this is correct or not? Like this is one of the way by which they have started the expression or not?
It has to be correct.
Yes, ma'am.
Yes, yes. So this is correct. Okay, but what do we see that in all the options A is present. इससे भी आपको hint मिलना चाहिए that your A is correct. ठीक है? Now, expression of new RNA polymerase. Obviously, right? अभी जब हमने बोला sigma is responsible for bringing different types of RNA polymerase, क्योंकि sigma बताएगा कि किस तरह का RNA polymerase आपका recruit होना चाहिए and आपका express होना चाहिए. So already we are saying in this case that bacteriophages, they are recruiting, okay, they are recruiting different sigma factors. तो sigma अगर अलग है तो obviously आपका RNA polymerase भी अलग होंगे, right? Now जब RNA polymerase अलग है तो obviously the hollow enzymes भी अलग ही होंगे. RNA polymerase आपका core हो गया plus sigma becomes your hollow enzymes. तो जब मैंने A and B right बोल दिया तो obviously C भी right ही होगा, correct? But then we don't have the different transcription factors. क्या है हमारे पास? In the prokaryotes, do we use transcription factors? Have I ever
No.
mentioned in the case of bacteria about the transcription factors?
No, ma'am.
No. तो इसका आंसर क्या होता है? बहुत ही सिंपल है, करेक्ट? अगर even if you don't know about the sigma, RNA polymerase, hollow enzyme, you should just remember that in the case of bacteria or the prokaryotes, we don't have transcription factors. We do have in the case of eukaryotes. So simple answer, simple logic. Option 4. ठीक है? So now do you see how much simple it is? Even if the question is so big, हमने कभी पढ़ा नहीं bacteriophage के infection के बारे में, but still it was easy. Okay, so this is how your 4 marks question comes. Okay, so the answer is 4th, A, B, C. Okay, now next question. So rifamycin and actinomycin. One second, I can't read. All this thing is coming. Rifamycin and actinomycin D are two antibiotics which are derived from the bacterium Streptomyces. Okay, so rifamycin binds to the beta subunit of the E. coli RNA polymerase and interferes with the formation of the first phosphodiester bond in the RNA chain. Whereas actinomycin D binds to the double-stranded DNA by intercalation. Okay, ये आपको पहले ही बता दिया रिफामाइसिन कैसे एक्ट करता है एंड एक्टिनोमाइसिन डी कैसे काम करता है. तो इससे भी आपको याद रखना चाहिए ऑलवेज रिफामाइसिन इज गोइंग टू बाइंड टू द बीटा सबयूनिट ऑफ आरएनए पॉल, ओके, एंड एक्टिनोमाइसिन डी इट बाइंड्स टू द डबल स्ट्रैंडेड डीएनए बाय इंटरकैलेशन. ओके, सो दिस इज हाउ इट इज गोइंग टू एक्ट एज द एंटीबायोटिक्स. सो इस क्वेश्चन से भी हमें दो नॉलेज मिल गई है आज, लाइक हाउ डज द रिफामाइसिन एंड एक्टिनोमाइसिन डी एक्ट्स. Now let's see what is the question, what is the answer over here. Sorry, what is the options asked? Which stages in transcription would you expect rifamycin and actinomycin D to affect primarily? Yes, कैसे होगा, क्या होगा आंसर?
Ma'am, one may be affecting initiation and other rifamycin. Three maybe, three option, third option.
Okay, what about others? Okay, time is getting over fast.
No, ma'am.
All three?
Two.
Two? Okay. Now what is the third option by the third person? एक ने 3 बोला है, एक ने 2 बोला है. Yes. Shruti, what you have given?
I'm thinking 1 second. Three, ma'am, 3.
Okay, all of you are wrong. Answer है इसका 1. See, I don't know how you people are reaching the third. See, rifamycin क्या बोला है? It binds to the beta subunit of E. coli RNA polymerase. जब आपका RNA polymerase ही assembled नहीं होगा, why? Because the beta subunit has been bound with the rifamycin. आपका RNA polymerase ही नहीं बना है, okay? So आपका पहला ही phosphodiester bond नहीं बना है. So what does it mean? है ना? See, we already said that the RNA polymerase is made up of beta, beta dash, 2 alpha and omega, and later the sigma comes. But if your beta has been bound by the rifamycin, तो RNA polymerase ही नहीं बना. जब RNA polymerase नहीं बना तो इसलिए आपका first phosphodiester bond नहीं बना in the RNA chain. So that is your polymerase binding, affects the RNA polymerase binding. Now, actinomycin, they have said that it binds the double-stranded DNA by intercalation. तो जब आपका DNA ही affect हो चुका है, how it is going to allow further transcription, initiation, elongation? वो आपको आगे बढ़ने ही नहीं देगा, तो आपका ट्रांसक्रिप्शन शुरू ही नहीं होगा. ओके, नाउ यू आर गेटिंग. तो वहां पे इनिशिएशन एलोंगेशन का सवाल ही नहीं आता है. अगर मेरे डीएनए में यहाँ पे मेरे पास इंटरगैलेक्टिक एजेंट्स है, आई कैन नॉट रिपेयर इट. अगर यहाँ पे मेरा ट्रांसक्रिप्शन इज रेडी टू प्रोसीड, बट आई हैव गॉट दीज inhibiting what to say intercalating things are present, I am not able to proceed further. तो ये वहीं पर ही inhibit हो जाएगा. Okay, now are you able to understand?
Yes, ma'am.
हाँ हाँ.
Yes, ma'am.
इसमें ऑलरेडी मेंशन कर दिया था that it is binds to the beta subunit of the RNA polymerase. So obviously उसमें बनेगा ही नहीं आपका RNA polymerase bind ही नहीं करेगा आपके ट्रांसक्रिप्शन साइट को, because RNA polymerase is affected. Okay. Now next question. ये तो सिंपल है. So the organization of an eukaryotic gene expressed at high levels in liver is dramatically represented below. So maybe it's not clear, I'll let you know. So it's a promoter of 5 kb, 5 dash untranslated region of 0.4 kb, exon 1 2 kb, intron 1 1 kb, exon 2 4 kb, intron 2 3 kb, exon 3 6 kb, and 3 dash UTR untranslated region 0.6 kb. So the size of the mature mRNA generated by the transcription followed by the normal splicing of this gene will be dash. Assuming that the mRNA is not polyadenylated, and 5 dash 3 dash refers to the 5 dash and 3 dash untranslated regions respectively. So what is the mature mRNA length? Yes. क्या होगा इसका आंसर? वेरी सिंपल गाइस. ऐसे से सिंपल क्वेश्चंस आते हैं 4 मार्क्स के लिए फॉर योर एग्जाम. यस, क्या होगा आंसर? ये अगर आंसर आ गया सो इट मींस यू नो transcription is understood by you guys. Okay. Yes, Akshaya, what will be the answer? तन्वी बताएगी.
13 kb.
Okay, Akshay is saying 13 kb. What about others? Tanvi, what is your answer? जो भी बोलेगा उसको उसका justification भी देना है, ठीक है? Yes, Tanvi, Shruti. Don't take this much time. 1 minute में आपको answer आना चाहिए. अगर 1 minute में आता है तो it means you have understood the concept and it is a correct answer. If you're taking more time to think about it, then it means definitely वो गलत होगा. कोई भी क्वेश्चन पढ़ने के बाद within 1 minute आना चाहिए. Yes. Shruti, Tanvi, pass.
13 kb. 2.
Okay, she is also saying 13. Tanvi, तुम बची हो.
Pass.
Okay, तो Tanvi pass. तो इसका मतलब वो बच गई है. Now, Shruti, justify your answer.
I'm confused about the intron part. I know that, not the intron part, sorry, the untranslated part. Introns get removed, but not sure whether the untranslated regions are there or not. So it's between 13 or 12.
हाँ, अभी 12 होता तो बहुत ही आसान होता ना. The thing is, answer is correct. The 13 is correct. Okay. Now, what your doubt is true that it should come. But आप ये भी तो सोचो ना. What does it say? It's an untranslated region. We are at the transcription level. Correct? So after promoter, these are the regions which gets transcribed till your 3-UTR. And as just now we have studied, during the elongation itself, your mRNA is getting spliced. यानी कि introns उसके remove हो रहे हैं. So obviously from 0.4 2 kb plus 2 kb, introns removed. Exon 2 4 kb plus again intron 2 removed. Then exon 3 कितना मैंने बोला था 6 kb and then last one 0.6 kb. So 2 plus 4 plus 6 12 plus ये आपका हो गया total 13 kb. ठीक है?
13.
हाँ. So the answer is correct 13. And जो doubt आया था कि like it should be there or not, it should be there. Already it is said that it is an untranslated region. वो तब का तब देखा जाएगा when they are asking whether the protein is having this region or not. So obviously we don't know. We have just mentioned that it is 4 kb, 6 kb. We are not asking how much long is your protein, correct? तो यहाँ पे क्वेश्चन है mRNA का, which is at the gene level, sorry, transcription level. So obviously this UTRs are also being transcribed. ठीक है? But we know that later in the protein it is not going to be present. Okay, so this is good. This is done. Okay, I guess only these questions were there. So there are many are there. It's just that you guys have to go through those PDF. That is like all collective questions from the previous question papers on the mole bio, which I have shared with you already. Okay, chalo, thank you guys. So we have done a lot today. We have completed with the transcription, and after this we'll be starting with the RNA splicing. Okay, so there are 3 methods by which RNA splicing is being done. So if you could get time, so you just go through about like how what is that intron 1s are removed with the help of, there are 3 types, means ribozyme 1, ribozyme 2, and then we have got these SNRPs. So those are your small nuclear ribonucleoproteins which helps in the removal of the introns. Okay, so these are the 3 methods by which intron splicing happens. If you could get time, just go through, or otherwise I'll be taking that. Okay. And about the evening class, I'll let you know, guys. Okay, थोड़ा सा मुझे checkup के लिए जाना है उसके. If I'm back, then we'll have the class. Okay, I'll message accordingly. Okay, any doubts? Okay, so I'll try to upload all those. So I have already uploaded 3. Just entering your email ID so that you can get those videos. Okay, chalo then. Bye bye, guys.
Thank you.
Bye, ma'am. Take care.
Thank you, ma'am.
Thank you.